right, is about measurement. And I want you to understand certain concepts. For instance, in a computer, you have 1 gigabyte of memory. It's equivalent to 1 billion bytes. Yeses and nos. Right, the bigger it is, the more files you can put on your computer. I think you understand that concept. So G is for gigabyte, your abbreviation. It's 9 zeros after the 1. So a milliliter is 1/1000th of a liter. That means there's 1,000 milliliters in 1 liter. If you take a meter, there's 1,000 millimeters in 1 meter, right? So liter and meter, one's the distance, liter is a volume. So a volume can be measured in, say, you know, if you take a cup, a standard cup has 240 milliliters. This measures out exactly 10 milliliters. If you were to put 10 milliliters of water in this, I usually have you do this in lab, then that amount of water would weigh 10 grams. Each gram of water is a milliliter. Okay, so it's a way of calibrating that. Now, I've got the videos for the microscope. Right, and then this is a microscope. You start with focusing with the 4x first, if we were to do it. In that video, I kind of go through how these are the objective lenses versus the lenses, and where the stage is, and where you have the where you put the slide that you want to look at. So, I want to be clear. I mentioned this in the video too. The ability of the microscope to remain in focus while the objective lenses is changed refers to parfocal. The field of view is the area seen within the ocular lens. Field of view, when you look into it, what do you see? That's referred to as the field of view. Depth of field is the focal length when viewing the slide, or how much of the slide is in focus. Okay, depth of field. If it's not in focus, then it's not in the depth of field. It'll look blurry. We'll use, if we were to do the lab, we would use the three thread. This is, if you were to view this under the microscope, you call this the 3-thread slide because there's 3 different color threads, and it helps you see depth of field. Right, the yellow thread is in the depth of field. The blurry ones aren't in the depth of field, right? Because that's what's in focus. So I expect you to know field of view, what you see through the ocular lens, parts of the microscope. So you won't be quizzed on that, but you'll probably see on the practical. You have to be able to label what's the ocular lens, what's the objective lens. Well, where's the stage, right? The stage is the very top item up there. That's what you put your slide on. The condenser, and you should know the fine and coarse focus knob. And where the light is, it's at the very bottom. You should be familiar with, at least for the practical, what a beaker looks like. A beaker are these items here, right? Both of those are considered beakers. It's right over here. You know, test tube's pretty straightforward. Erlenmeyer flask has that triangle look. So you might get, I expect you to know all 5 of these. Test tube, pasteur pipette or pipette, looks like that. I don't think I have one in that box, but that's a pasteur pipette. You put a bulb on the top of it and it can suck things up. Graduated cylinder, right? Right, this is a 10 milliliter graduated cylinder. You can accurately measure out 10 milliliters. pH, I just kind of quickly go over this. You know, phenol red we'll be using in different labs. Is considered what you call a pH indicator. And if it's the pH is above 7, it appears red. If it's below 7, it becomes yellow. I'd expect you to understand pH is the measurement of the hydrogen ion concentration. So it always refers to water when you say, what's the pH of a solution, right? It's an aqueous solution or water. And acid means it's below. If it's something you add that causes the solution to become acidic, right, or yellow like this, right, because it's below 7. If you add a base to water, then it becomes basic or alkaline and has a pH above 7. Okay? So you could use things like indicators to know if it's acid or a base. If it's an acidic solution on this side yellow or a basic solution on the other side. It really doesn't know. You really won't know if it's pH 7, but it'll be below or above. A buffer maintains that, right? Use this PowerPoint mostly. Watch the videos to help you understand it, right? But like I expect you know the pH scale goes from 0 to 14. Right, I already talked about an acidic solution, basic solution. That describes the solution. And acid is a noun. It reduces the pH to make it acidic. A base, something when you add to water, makes the water basic or a basic solution above 7. Okay. What I'll do, we'll talk about the macromolecules in the next lab. Quiz number 1, though, covers this lab and next week's lab, right? So if you look at the